so that it activates the signaling pathway of TLR. As mentioned before, the risk factor, one of the risk factor is genetics. MHC class 2 is one of that. If we upregulate it, it will produce molecules that are vital for the activation of T cells. And for your information, T cell is one of the cells of immune system which gonna attack beta cells, and without beta cells, we cannot produce insulin for our body, right? And when the upregulation happens, it's gonna produce type 1 interference, and this type 1 interference is gonna help the T cell to survive better, and it's gonna attack the beta cells better. And so our group think that, what should we do with this? We think we're gonna plug this one, especially TLR2 and TLR4 by using the poly-IC. So why we gonna plug this one? First, we don't have to inject anymore because injection is very painful, you know, right? And secondly, we're gonna plug this from the beginning, so the T cell cannot kill beta cell anymore, so that we can save more beta cells. Yay. And let's move on to the very last part, biological model. Can you see it? Very cute, right? It's the mice. There are several reasons we choose this to become our samples. First, we can inject Coxsackie virus into them, and this virus is taken from the diabetes patients, and we're gonna inject into the cutie mice so that we can simulate it as diabetic in mice, and yeah, and we're gonna simulate it by killing the islet cell inside them, and moreover, it has 13 TLRs. Instead of in our humans, we got only 10. And if we increase the TLR3 and TLR2 and TLR4 expression, well, the output we have is going to be this significant correlation with HbA1c. And with the result of HbA1c, we're going to know, we can analyze the plot of the mice. And last but not least, it's very cheap. Yesterday, I have a tiny research about the cost of the mice, and I got the answer from the University of Adelaide. It's gonna cost only $9.87 per cage. So that's if we have any mistake with the experiment, we can buy a new cage, which is not gonna cost a lot. And moreover, we can conduct in a large amount of mice so that the result will be better. So that's all for our presentation. Sorry for any flaws we have made, and we will be very happy to receive the feedback from you guys, and thank you for your listening.